Hello, welcome back. This mini lesson covers neuroglia, which are the supporting cells of the neurons. I like to think of the neurons as like the main players, almost like the rock stars of the nervous system. They are very picky about their environment. They have the electricity to send these action potentials and communicate all over the body. Where the neuroglia are like the roadie crew. They are behind the scenes. They help to support the neurons. They make sure they have everything that they need taken care of because the neurons are very picky and specific as to what they want. So this mini lesson is going to be all about the different types of neuroglia that are found in the peripheral nervous system and the central nervous system because they are different depending on where in the body we're going to be taking a look at these neuroglia. So we have this diagram here. I think in your book it splits between the two. So I've just cut out the CNS portion. So when the neuroglia are found in the CNS, there's four different types. We have the ependymal cells, the microglia, the astrocytes, and the oligodendrocytes. So I'm going to go through them in order, right? So in order on the notes here. So the first one listed are called the ependymal cells. So these are neurons that are found lining the hollow spaces in the brain. You do have hollow spaces in your brain and spinal cord. They are called ventricles in the brain and the central canal in the spinal cord. So there's fluid filled. So these ependymal cells kind of line those cavities, and some of them are ciliated, which means they help to circulate the cerebral spinal fluid, which we'll talk more about when we get into 232. They do a little bit of production, but not as much as the main producers of CNS, CSF, cerebral spinal fluid, in the CNS. Their major job is lining those hollow spaces and helping with the circulation of the cerebral spinal fluid. The next one on the list is the astrocytes. These are the busiest, if you will, of the neuroglia. They have a lot of different jobs. Astro meaning star, so I guess they kind of look like a star cell. So that's the early anatomists thought that's what they looked like. So their major jobs listed here. It's also listed in the notes, or you can list it in the notes in the book. From the book, they make the blood-brain barrier, which again, we'll talk more about that when we get to the brain anatomy. They help create a three-dimensional framework within this neural tissue to help support and guide the direction of the axons of all of the different neurons. They help with limited repair of neural tissue, and it's more often that they inhibit repair than actually help with repair. They guide neuron development, part of that framework job listed earlier, and they help to control the interstitial environment, so regulating the amount of neurotransmitters, helping to recycle neurotransmitters, making sure the ion concentrations are accurate to help with the physiology of action potentials. So those are the astrocytes. The next ones are the oligodendrocytes, so that's a big mouthful. They help with what we call myelination. I'm going to show you myelination here in a couple slides. So myelination helps to insulate the axons of neurons, and it helps to speed up the propagation or the sending of these action potentials, these nerve impulses, down the axons. They do also help to support and give structural framework to the neurons in the central nervous system. Okay, so those are the neuroglia of the central nervous system. So ependymal cells. Oh, sorry, one more. I forgot about little microglia over here. So microglia are the phagocytic cells of the brain and spinal cord. So they help phagocytize debris. They're like the cleanup crew. They get rid of waste and cell debris and can recognize pathogens. There we go. So that's all four mentioned. So ependymal cells, microglia, astrocytes, and oligodendrocytes are the CNS of the neuroglia of the CNS. Okay, so here's a picture kind of showing how all of those cells would be organized within, in this case, the spinal cord, because they're showing the central canal right here. Okay, so that's going to be a fluid-filled space filled with cerebral spinal fluid. We can see ependymal cells are completely lining that space. Here we can see an astrocyte doing its job of helping to support the axon. That's the blue band kind of in the middle. And then we can also see it's completely covering that capillary. So we don't get anything leaving the capillary unless it can also pass through the membranes of those astrocytes. So that's basically what the blood-brain barrier is. So here we can see a microglial cell just kind of cruising around looking for cell debris or bacteria or anything that it can gobble up. And then here's the oligodendrocytes. So they send out their processes and they wrap and wrap and wrap and wrap around the axons until you get this thick layering of multiple layers of their processes. And this is what we call myelin, or the myelin sheath. Okay. Another thing I wanted to mention in this picture is the difference between gray matter and white matter. You've probably heard of those terms before, kind of in reference to your brain, which is true for both brain and spinal cord. Gray matter is indicative of where you have more cell bodies, because cell bodies are not myelinated. And so upon dissection, where you have higher concentrations of cell bodies, the tissue looks a little bit gray. Where you have a lot of myelinated axons, myelin is primarily adipose tissue. It's fats, phospholipids, and fats are white when you have a whole bunch of them together. So where you have higher concentrations of myelinated axons, the tissue appears lighter in color. So that's how we get the difference between gray matter and white matter. All right, so those are the neuroglia of the CNS. So now we have the other half of that chart with the two neuroglia that are found in the peripheral nervous system. So we have one that are called satellite cells, and they just kind of hang out around the cell bodies, very much like the astrocytes in the CNS. So they regulate the gases, they regulate neurotransmitters, the nutrients, the ion concentrations. They're kind of the real, housekeepers of the peripheral nervous system neurons. And then we have, so those are the satellite cells. That was a bad circle. The satellite cells. And then we have the Schwann cells. So the Schwann cells are the kind of comparable to the oligodendrocytes of the CNS. So Schwann cells are what do the myelination in peripheral nervous tissue neurons. So here's a Schwann cell kind of starting here, and it just keeps wrapping itself around, and it will wrap and wrap and wrap and wrap until you have multiple layers of the Schwann cell just wrapping and wrapping around, and that's what's going to give you the myelin. So we can see that here. When we talk about the propagation, there's going to be a video on propagation of action potentials. This is where the oligodendrocytes and the myelin cells the Schwann cells are going to come into play when we talk about how they can increase the speed of that action potential running in this direction. Okay. And the last thing I wanted to talk about in this video is in the peripheral nervous system, not in the CNS, there's actually inhibitory mechanisms that prevent neuron repair in the central nervous system, brain, and spinal cord. But in the periphery, there's a limited repair, and those Schwann cells help with that. So let's say you have a neuron and it's been severed in half, okay? You're gonna have macrophages come in and clean things up, and you're going to have inflammation, you're going to have try to get repair, but what you'll have is this proximal stump, right? So the neuron is cut and severed, but the Schwann cells are kind of still hanging out. We have your connective tissue sheath, and then we have the Schwann cells hanging out just inside of that, and they can regenerate. Neurons typically can't grow completely new neurons, but the Schwann cells can make more Schwann cells. They'll reconnect to that distal stump of the rest of where the neuron was going, and then this proximal stump of axon will just continue to grow through that new repaired tube and go and make the connection to the effector of what it used to be. So there was a story, I usually tell this story. It's not my story, I can't claim it's my story. We had another part-time instructor who he told the story, and I like to carry on the legacy. So he had a cat, right? I'm a horrible drawer of animals, right? So the cat got its tail caught in a door, and it didn't lose the tail, but what happened was the tail kind of got broke, and then after the point of damage, it was just floppy. The cat had no control over it. So it was nerve damage. But then after so many days, weeks, months, I think it was on a period of months, the dangly bit got less and less until finally the cat got complete control of its tail again. So he was able to witness this slow progression and regaining of control of that tail as that peripheral repair actually took place, which is pretty cool. But this is limited to just peripheral neurons. This is not something that you would see take place in the brain or the spinal cord. Okay, That's it for now. I'll see you next time.